All right, time to go teach. Things that can affect how well an enzyme works would be temperature, pH, the concentration of our substrate or our enzyme, and inhibitors. So we're going to talk about temperature, pH, concentration, and inhibitors in order. So the first one was temperature. Here's how it goes. If we look at a little graph, activity, this peak would be where it's an optimal activity. This is where the enzyme is working at its absolute best. Temperature at its coldest to its warmest. And what you can see is it does decrease as we get colder, that activity. It goes down. And the reason that the temperature will go down is because temperature is a measurement of how fast molecules are moving. If molecules are moving slower, that means that the substrate and the enzyme will come in contact less frequently. And if they come in contact less frequently, you're going to have less activity. But you'll also notice that when we talk about getting too hot, it drops off pretty quickly and it goes straight to zero because heat, while it will actually cause the molecules to move faster, it also breaks hydrogen bonds. All of the secondary structure and a lot of the tertiary and quaternary structure is due to that hydrogen bonding. So if you don't have hydrogen bonding taking place, it's not going to have the proper shaped active site. The active site is actually going to unravel a little bit, and that's going to make it where the substrate no longer can fit. If we change the shape of the active site, the enzyme can't function. That active site has to be the proper shape. Otherwise, the proper substrate can't fit inside the active site. And that's a really big deal. So if you get too hot, the enzyme will cease to function. This also happens with pH. You can see here we have like this optimal activity again for that pH, and you also see though on both sides of it, it ends up dropping off. Now this is not exactly at pH of 7. I don't want you to think that enzymes work in a neutral solution only. That's not the case. Inside your blood, your pH is approximately like 7.4. The enzymes in your blood work at the pH that your blood is at. The enzymes that work in your stomach though, they need to work at a pH of approximately 2. That's a really big difference. So the optimal pH of an enzyme is specific to the enzyme, in the same way that the optimal temperature for an enzyme is also enzyme specific. Some enzymes work at a very large range of temperatures. Some enzymes only work at a very small range of temperatures. Same thing with pH, where it's a very specific thing to that specific enzymes. Some enzymes only work at a pH of 2, and that's it, a really narrow graph, and they would not work at a pH of 1 or at a pH of 3. Some enzymes are able to work at a pH of 5 to 8, and so it's going to have this wider bell where you have the enzyme working at best at approximately 6.5. It depends on the enzyme. All enzymes have their own specific optimal temperature and their own specific optimal pH. So you can't say what that pH is by this graph. We don't know what that pH is. From the previous graph, we don't know what that optimal temperature actually was. We just know that every enzyme has an optimal temperature and an optimal pH. And when we go outside of that, they do not function as well. One of the big things that can happen is this thing called denaturing. That means that the enzyme has unraveled because we're breaking down the interactions at the secondary, tertiary, and if it has it, quaternary level. If we break down all those interactions, our polypeptide chain, well, it's just going to be a polypeptide chain. It will not be a protein because there's no way it can function. And that means it will no longer be able to do what it needs to do. So an enzyme that has been denatured, that means we have broken it down to just really its primary structure. The only thing left is we have a polypeptide chain with a specific order of amino acids. We have messed up all of the other levels of structure, and that means that we have just destroyed all of its functionality. This happens at high temperatures, and it also happens if our pH goes too high or if our pH goes too low. So that is something that can happen to make it so that our protein stops functioning. As the substrate concentration increases, that means that you're going to end up with more substrate coming in contact with the active site of another enzyme that's empty. And that's great. But after a while, as we keep adding more and more and more substrate, we're going to get to the point where there are no longer any empty active sites. All of the enzymes are actively involved in the reaction. And so that means that those substrates are just going to be sitting until an active site ends up being open. And that's what we call enzyme saturation, where no matter how much more of our substrate we add, the enzymes can't work any faster than they're already working. They're all working as fast or as quickly as possible. That would be called enzyme saturation. There is no empty enzyme that can act faster. So that is the different ways of measuring activity. And now we're looking at inhibition. What is inhibition and how does inhibition actually work? Well, there are two types of inhibition. There's competitive inhibition and there is non-competitive inhibition. So let's check that out. First, competitive inhibition. In competitive inhibition, we still have our substrate, and now we have our competitive inhibitor. Now, the competitive inhibitor, you can probably tell it has a specific shape, and that specific shape fits in the active site. So it's going to compete with the substrate to go into the active site. If the competitive inhibitor goes into the active site, then that means that our substrate can't fit, and our reaction will not take place. The enzyme, for the period of time that that competitive inhibitor is present in the active site, it can't function, it cannot work. It is effectively turned off. Now, this is not a permanent thing. The competitive inhibitor will release, and then the active site will be open again. But for a short period of time, the inhibitor is going to make it so that the enzyme cannot actually have a substrate come into it because its active site is already occupied. And if the active site's occupied, the substrate can't fit. That's just the way it works out. That is competitive inhibition. That's one way that an enzyme can effectively be turned off. Another way that an enzyme could effectively be turned off is called non-competitive inhibition. And again, we got our substrate right here, and we have our non-competitive inhibitor. But I want us to also pay attention to, we have this other site down here. It is called an allosteric site. And that is where the non-competitive inhibitor is going to bind. So the non-competitive inhibitor, it doesn't go to the active site. It goes to the allosteric site. And when it fits into the allosteric site, it causes a conformational change, a change of shape in the active site. So now our substrate doesn't fit. And since our substrate doesn't fit, again, the enzyme is effectively turned off. Just like with competitive inhibition, non-competitive inhibition is not permanent. That non-competitive inhibitor will release, and when it releases, the active site will change its shape back so that the substrate can fit inside of it. So it doesn't mess up or destroy the enzyme, it only turns it off for a short period of time. It only is off while the non-competitive inhibitor is bound to that allosteric site. And this has some interesting effects when we look at our activity of our enzyme. So for the activity of our enzyme, you should probably remember this is our substrate concentration graph that we just showed a little bit ago. And right here, this is where the enzyme was completely saturated. It could not act any faster because the enzymes had no free active sites to take in more substrate. They were interacting with the substrate as quickly as possible. It's almost like if ever you've gone to the grocery store and all of the cash registers, there's like a big line in each one. Well, the people that are working the cash register are working as fast as possible. They're trying to get through that line. They can only ring people up as quickly as they can ring people up. So it's the same way for the enzyme. It can only work as quick as it can work. If we look at this graph, when we have a competitive inhibitor, the key is the more substrate we put in, that means that the substrate can find or come in contact with the enzyme more frequently because it will beat out the competitive inhibitor. Because they're both competing, it ends up that if there's more of the substrate, it will come in contact with the active site more frequently than the inhibitor. So it can still end up getting to enzyme saturation. It can still make it to its peak rate as long as we continuously add more and more and more and more and more substrate. But non-competitive inhibitors, since they're not going to the same site, they end up where there's a peak rate that can have, and it doesn't matter how much more substrate we add. We can keep adding more and more and more substrate, but these enzymes, well, there's always going to be a certain amount of them that are going to be turned off. And since the non-competitive inhibitor is not going into the active site, since they don't compete with the substrate, that means that it doesn't matter how much more substrate we put in, the allosteric site will always be open. It will always be available just to that non-competitive inhibitor. So it will end up reducing that peak rate where we have enzyme saturation. So this cannot be overcome in the same way as competitive inhibition can be overcome. Competitive inhibition, theoretically, you just keep adding more substrate. You can actually overcome that inhibition. But non-competitive inhibition, that can't be overcome. And this is one of the ways that our body uses enzymes, but also uses inhibitors to maintain homeostasis. The inhibitors help keep the enzyme activity in check so that the reaction rate is what our body needs and not just always on or always off. So in summary of all that, enzymes, what do? Well, they speed up chemical reactions. That's their job. Enzymes are to speed up chemical reactions. How do they do it? Well, they stabilize that transition between reactant to product. And because they stabilize that transition between reactant to product, they lower that activation energy. That is how an enzyme works. Next, that has a very specific location where that process is going to take place. That takes place in the active site of our enzyme. And the active site of the enzyme is specifically shaped so that our reactants can fit into that active site. I only showed an example of hydrolysis, taking one substrate and breaking it into two products. But I could have actually shown dehydration synthesis, where we're going to take two reactants, put them inside of our enzyme, and they will become one product. We could have either thing take place, all having to be able to fit within that active site. That active site shape is really important. Enzymes are extremely specific for the substrate that they work on. And their activity is dependent upon a few different factors. Is the pH and temperature the optimal pH? Is it the optimal temperature? What's the concentration of our substrate? And what inhibitors are present? What molecules are out there that is going to cause either non-competitive or competitive inhibition for our enzymes? That's it for this time. Till next time, be awesome, stay awesome.